Good morning, dear students. Today I am going to teach you a very interesting topic in genetics, which is known as Mendelian genetics. As you all know, Mendel is known as the father of genetics. So we have to know all the things about the Mendel, how he developed the genetics, what are the materials he used for his study. As we can see here, is Mendel. Mendel, as a Mendel born in 1822 and he became death in 1884. And Mendel is responsible for the law that governed the inheritance of traits. As we know, the inheritance of traits means transfer of traits from parent to progeny. And Mendel, here the history of Mendel. Mendel born in 1822, Austria. Now it is situated in Czechoslovakia. He has joined 1847 as a priest. 1851 higher study he went to Vienna University. 1854 he works as a science teacher and 1884 Mendel became death. Before his death, he started the study in the pea plant in 1856 and cultivated to over 28,000 pea plant in his garden. He found that the plant offspring retain the traits of parent, means they have similar character according to their parents. In this, he had developed the law of inheritance and he presented his finding in the Natural History Society of Brunn in 1865. His paper was entitled as Experiment in Plant Hybridization that was published in 1866 and in German language. Mendel's work was not recognized until the 20th century, but during 1900 there are three scientists, Carl Corens, Hugo de Vries, and Heinrich von Tismark. He rediscovered the law of Mendel. So, after the discovery of law of Mendel's by three renowned scientists, Mendel, known as father of genetics, and site up here we can see the experimental garden of the Mendel where he grown the different color of pea plants. Here the microscope that is used for his study by Mendel. Here the means before Mendel proposed the particulate inheritance. He means particulate inheritance means that a particular particle that is responsible for the inheritance of a trait from parent to progeny. These particles was told to by Mendel as a factor because Mendel don't know about the gene. He told that gene as a factor. After that, there are different terminology that is used in genetics like monohybrid cross means here the involves crossing two parents that differ for a single trait like flower color. Dihybrid cross means the crosses involve two parents that differ for two traits. Alleles means the alternative form of a gene that is known as alleles. Alternative form of gene means like dominant or recessive. A single character has two form like height of a plant that has two form tall and dwarf. One is dominant, one is recessive. Dominant character means the character that is expressed in the heterozygous condition, means the character that expressed in the hybrid, that is known as dominant. The characters are the gene that is not expressed in the hybrid, that is known as recessive characters. Genotype means genetic architecture, a genetic combination of a trait like capital RR, small rr, capital T, small t. These are known as genotypes. Phenotype, the physical appearance, means whatever we can see by on their naked eyes, that is known as phenotype, like red color flower, white color flower. Here homozygous genotype, the genotype that has two similar allele at the correspondence loci, that is known as homozygous genotype. And heterozygous, when the two allele are different at correspondence loci, that is heterozygous genotype. Here we can see phenotype purple genotype capital PP purple, capital P small p heterozygous, that is also purple. Purple small pp white. Here means the dominant character is purple, so it is expressed in both heterozygous and homozygous condition. A recessive character is non-white, that is expressed only in the recessive homozygous condition. Mendel's pea plant experiment. You can see the pea plant is Hindi is known as matter. As why Mendel selected pea plant? But there are very logic, so many logics, so many in interview. This question commonly asked why Mendel selected pea plants. Because pea plants, as we you have seen, it can take only small place to grow. It produce lot of offspring. Offspring is progeny. It produce pure plants when allowed to self pollinate. It can manipulate several generation. It can be artificially cross pollinated. It is bisexually, many traits already known. Above all, it is easy to grow. Here we can see the pea plants. Mendel mainly, what are the experimental method that is used by Mendel? Mendel mainly used hand pollination in the flower. He cut the flower snipped stamen and to take prevent the self pollination, and he traces traits in the several generation. Here we can see the experimental Mendel how he performed the experiment, remove the stamen from the purple flower. Here after that the transfer of pollen from stamen to white flower, pollen carpel of mature. We can see the pod here, mature pod we can see. The, after that plant seed are planted in pod. Here we can examine the plants. How Mendel began experiment? How Mendel began self? Here we can see Mendel produced pure strains by allowing the plants to self-pollinate for several generations. He allowed plants to self-pollination for several generations. There are different character studied by Mendel like seed shape, seed color, pod color, flower position, stem length. Here Mendel's experiments result. We can see the result. He has predicted all the results in monohybrid 3 is to 1 phenotypic ratio, 1 is to 1, 1 to 1 genotypic ratio. In dihybrid case 9331 and 1 to 1 242 1 to 1. You can see here the real time monohybrid. After that, in another generation here, the parents P1 using cross F1 the generation that is obtained from crossing between two parents, F2 is the generation that is obtained from the selfing of the F1 generation. Here following of the here we can see P1 generation, P generation after F1 generation after selfing F1 we can see the F2 generation. Crossing between two floor plant here we can see the result. Monohybrid cross means the cross where two parents used in cross that differ for a single traits that is known as monohybrid cross. Here we can see alleles capital I smaller. There are two round seed and wrinkled seed after crossing. Genotypic ratio a phenotypic round after we can see genotypic ratio 2 1 to 1 phenotypic ratio 3 to 1. Here up homozygous dominant homozygous we can see the here F1 seed safe we can miss initially we can predict the monohybrid class and their generation. Monohybrid cross review heterozygous with heterozygous cross offspring 25% homozygous and 50% heterozygous 25% homozygous recessive offspring F2 generation. Why do we the P looks like here we can see the P plants. Now the test cross means crossing of the F1 to the recessive parents is known as test cross. Test cross is mainly used to know the genotype of particular parents. F2 monohybrid cross that is mainly used for the test cross analysis because test cross is mainly used to know the genotype of the parents. Here next monohybrid cross review. Here monohybrid cross review homozygous heterozygous 50% of spring. Here the Mendel's proposed two law of segregation and law of independent assortment. Law of segregation, the two allele of a genes remain separate and do not contaminate each other during F1. But during gamete formation, they segregate and pass separately into different gametes, so maintaining their own purity. Here, law of segregation during the formation of gametes means when the gametes form, the alleles going separately and maintain their own purity. So, law of segregation is known as law of purity of gametes. We can see here two parents cross purple flower, white flower. Here F1 purple flower. After F2 generation we can see 3:1 ratio. Law of independent assortments, each allele pair segregate independently of each others without 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 disturbing anyone, and assort independently and pass to save them. This law that is used in dihabit cross. Dihabit cross, we can see dihabit cross means that traits the parents that differ for two traits like seed shape, seed color. Here we can see dihabit cross, capital RR here, round wrinkled seed, round yellow and round and wrinkled yellow and green. Capital R round, capital Y yellow, small R wrinkled, small y green. Here all are possible combination we can see 9:3:1 ratio, phenotypic ratio, round and yellow, 3 round and green, wrinkled and yellow 3, wrinkled green 1. Summary of Mendel hypothesis. Gene can have alternate form that is known as alleles. Each offspring inherit two alleles, one from each parent. Means the two parents use in cross, they pass single allele to their progeny. If the two alleles differ, the dominant allele expressed recessively remains hidden unless the dominant allele absent. The two alleles for each other segregate during gamete formation, that is known as Mendel's law of segregation. Different characters have no effect on each other. Each allele pair segregate independently of others, that is known as law of independent assortment. Here we can see the parallel relationship between Mendel factor and chromosome. Means Mendel factors now known as gene. Here the rediscovery of Mendel's, several scientists focused to identify the basis of inheritance. William Sutton and Barberi proposed the chromosomal theory of inheritance. Chromosomal theory of inheritance correlate the means the chromosome and factors, like factors segregate, chromosome also segregate. Factors are gene present in chromosome, how the chromosome behavior in cell division, same like factors are gene behavior in cell division. There are different variation we can see in the Mendel's, like incomplete dominance, codominance, multiple allele, epistasis, and polygenic inheritance. As we know, Mendel's in F1 generation, dominance, pure dominance characters occur, means the character that show expression that in dominance character. But some variation, we can see the deviation from Mendel, like incomplete dominance. Here we can see incomplete dominance and codominance. Incomplete dominance, F1 hybrid had appropriate appearance somewhat in the between two parents, like when one parents current red, other is white. The appearance means pink color appear produced in F1. That is the incomplete dominance, means one character not showing complete dominance. So that is known incomplete dominance. Here we can see red, white by F1 generation. Here pink color. 1 to 1 ratio is 1 to 1. Both genotypic here we can see red and white, therefore generation pink generation. Codominance, when both the allele of a gene express in itself in upon generation codominance, we can see the cow cattle where the different patches in the skin of colors like red, black, white. That is due to the codominance. Codominance means both allele going to express their character in a one. Blood group of human, as everyone you know, there are different type blood group: A blood group, B, AB, and O blood group. Due to the codominance means all the alleles express their itself in that code. Next concept is lethal gene. Lethal gene, the gene that cause the death of an individual, where that particular gene present in their appropriate genotype before the adulthood. Means if that gene present in a particular individual, that individual cause death of before the adulthood. Lethal gene is this gene present in every individuals and plants like me and other, but this is always present in recessive homozygous condition. As we know, recessive allele only expressed in homozygous condition, otherwise they are masked by the dominant allele. So this condition never came, is all the individual become death. This is discovered by 1904 the Vaigyanik Delcunot in mouse. He observed yellow color is dominant, but he crossing two yellow mice. We got only 2:1 ratio. When he crossed yellow with recessive wild type gray, found all are yellow. Concluded that mice were heterozygote. Lethal gene. Later, he suggested that homozygosity for yellow is lethal. That the individual dies and Here recessive lethal we can see yellow crossed with yellow. Here gamete fertilization after that. Here we can see F1 generation. This date yellow become death because this yellow the gene lethal gene present in yellow, so we become death. Pleiotropy. Pleiotropy means there are two terms. One is polygene, one is pleiotropy. Polygene means a lot of gene governing single trait. Pleiotropy means single gene governing more than one trait. That is pleiotropy. Here, pleiotropy we can see impact of single gene or more than one character, like sickle cell anemia, like like the QPM quality protein match. The gene is known as OPEC2 gene that is called shrinkled of grain, and that increase the lysine and tryptophan content. Means two characters that is response single gene that is known as pleiotropy. RBC here sickle shape that is called many